Ho ho! Marcel, I love you. However, I'm about to do some microbiology labs, so make sure you bring your pets out of the room before you begin. So I'll be right back. Okay, now that my good friend Marcel is gone, let's go ahead and get down to business. And the first business is safety. And so again, you don't want your pets around, right? We're talking about microbes that can get you or your pet sick, and you certainly don't want these tracked around your home, your office, or wherever you're doing this. And so that's where now I'm fully suited up, right? I've got my lab apron, which came with the kit, a pair of gloves, and my goggles. And so there is some flexibility with this. With the gloves, no flexibility. You're always wearing gloves in this course when you're working with microbes, okay? In terms of the goggles, what you'll find, depending on the humidity levels, et cetera, in your lab, that sometimes these will start getting fogged up, right? And if you are getting fog on your glasses, then you really can't do the microbiology lab well. And so when I conduct microbiology and when people are doing this in person, if they have a pair of glasses like mine that are big enough, and you see they're not tiny, right? This is good enough for the microbiology lab. If you feel uncomfortable, certainly use the full frame, but I'm going to go ahead and just use my glasses because one thing I'm not going to do is I'm not going to be putting the plates up. Whenever you're working with microbiology, you always work with your hands manipulating things down close to the surface. If you ever drop anything and you're looking at it, it just drops right there. Okay? So whatever your comfort level is, use that, but you are authorized to use glasses if they're pretty big. You can also use safety glasses that you might buy for doing some workshop or something like that, some kind of resistant glasses. So in terms of the lab apron, there's nothing special about this, and actually I'm not a big fan of this lab apron. It will get the job done, but what you may want to do is you may just want to have a set of clothes that you put on whenever you do microbiology, and then when you're done, you can go ahead and launder those clothes. Okay? But the key thing is, whenever you are doing microbiology, the stuff that may come in contact with it should never be something you then walk around your house with. Okay? So that said, let's go ahead and get to the Ubiquity of Microorganisms lab. I've got my lab printed out in front of me, which is what I want you to do for every lab. Do not use your computer, right? Because you'll be tempted to touch things. If you get this contaminated, well, you can put this in a bleach bucket and kill everything and throw it away and print out another one, right? You contaminate your computer, then you're contaminating your computer. It can transfer to other people. You can get sick. We do not want that. Okay, so now when I say, you can get sick, I'm talking about if you accidentally contaminate something. If you pay attention, if you are cautious the entire time, there should be no problems. However, sometimes accidents do occur, right? So we want to minimize that, and ideally you won't spill anything, but if you do, you certainly don't want it to be on your computer keyboard, right? So what we have here are the plates that have been incubating for 48 hours. And so I've already cleaned this down with bleach. And so I'm going to take my plates and I'm going to put it out here. I have just touched the plates, and actually the aroma that is coming out right now is very microbial, right? And so all of the foul smells that humans give off come from bacteria, whether it's body odor, whether it's feces. Those are from the microbial activities with waste products volatilizing into the air, and you smell that. Okay? As a quick aside, whenever I smell something particularly nasty, which in this class you will smell some nasty stuff, I'm always reminded that actually those are molecules which are in the air that are then going into my nose, binding to receptors and sending nerve signals to my brain, and then we perceive that as a smell. So you actually are kind of tasting, right, those particular molecules. Okay? However, just because it smells bad doesn't mean that it is bad. Okay? So when we look at these up close down here on the tray, what we see is that, for example, the phone had the least amount of growth, but we see are some nice colonies, right? We see all of those spots. Whereas the soil, essentially there's bacteria all over the entire thing. Wonderful. And that's why we did soil, is to make sure that we got something. So now that I have touched these plates, it is really important for you to understand that these gloves we consider contaminated. Now, they really aren't, because nothing should have gotten out, but for strict microbiology rules, if you always do things the same exact way and you always treat everything like this is contaminated, then you will be safe. Okay, so part of the requirements for the lab is to analyze these. And what I recommend doing is first having pictures taken of them, some photographs. You need to upload them into the website anyway, as well as the discussion boards, so why not do that early on? However, how am I going to take a picture using my phone or a camera when I'm not now allowed to touch anything that is not right in here? And so this is where I really encourage you to have an assistant. Not Marcel, as cute as he is, and he does have opposable thumbs and fingers, right? I don't want him around the lab area. So instead, I'm going to go ahead and have Greg join me. And so Greg here is going to go ahead and take photos of these. Now, if we look at this, we can see that there's a little bit of shine there, but if I take this off, the photo is so much clearer. And so go ahead and take a photo of that. Take a photo of the next one. And get in nice and close. Okay. And now let's go ahead and take a photo of it on this. So you won't be able to see it on your screen, but Greg is going to get in nice and close and take a picture of that and trying to avoid a shadow on that. So that looks great. And then we're going to do the nose. Get in nice and close, a little closer. There we go. Great. All right. Thank you very much, Greg. And so now we have those photos. So let's talk about what we are looking at with these particular plates. So for each one of these, right, the lab manual comes as is saying to never open the lid. And now what's the first thing I did to show you? I opened the lid. Well, A, things that I do are for the camera so that you can see them, but B, you can open the lid if need be. Like right now, there is condensation, and so there is a film. So if we were to try to take a photo through that, I would be able to see hardly anything, okay? And then I wouldn't be able to grade your lab very well. Same thing, let's see, with the nose, we can at least see some. That's why the lab manual suggests you do it upside down, which is good for plates like this, but for plates like this, sometimes it's a lot harder. And the material or the information you get out of that is very different from the information you get out of looking at it from that way. And the other thing we'll look at when we see a photo is that the background matters. So holding it like this, right, we did that to show you that Greg was taking the picture, but what we don't want is this blue background or some type of busy background. You want a nice clean background, which is then why I had him take photos of them on this tray right here, so that there would be that background which is nice and clean. It doesn't have a pattern. So for people that are tempted to take a photo of it like on their granite countertop, Silex countertop, yeah, it looks cool, but the problem is when students submit that, then there's all these patterns and little specks in it, and it actually looks like bacteria, okay, which we want to avoid. So, so the things that we're going to do with this is we're going to analyze the different colonies, okay? And so let's go ahead and look at the colony morphology drawings in your lab manual and talk about the expectations for what you enter into the data table. So what we have here is the things that you need to put in. In addition to shape, margin, elevation, and size, you're also going to put in the color as well as the smell. Okay, what did it smell like? Nothing? Sewage? I don't know, but it wasn't good. You know, you can use terms like that. That's fine for this type of microbiology class. However, for the shape, you need to be very specific. Okay, so this is when you look at the overall colony. If it's just a circle, then it's circular. If it looks like this, it's rhizoid. If it's kind of circular, but it has protrusions sticking out, kind of like this, that would be irregular, right? And so then the margin is, if you look at this region really close, right? When you zoom in your picture, what does that look like? Is it just flat, in which case is it entire, or does it have big lobes coming out, lobate? And then the elevation you need to look at kind of from the side, and then the size is all relevant, all relative. And so if you have a whole bunch of colonies that are tiny, just say that they're pinpoint. If you have multiple colonies of different sizes, then maybe the biggest one is large, the smallest one is pinpoint, and the others are in between. Don't get worked up over like, oh my goodness, is it punctiform or is it small? just use your best judgment, okay? That's what we're looking for. And when it comes to other things, if it's really close, right, like if you give me, if you say curled and I look at it and I say, well, actually that looks like lobate to me, you're still going to get the points because you guys are just starting out, and the difference between these two, I really don't expect you to know. Now, if it's filamentous and you give me something like entire, then I know that you didn't quite understand what was going on and you made a mistake, and then unfortunately you won't get the points for that. So again, if they're really close, then you did your job. If I look at something and it looks circular and you said it was filamentous, then obviously there's some kind of big disconnect. Okay? So let's go ahead and for this, take a look at the individual pictures that Greg just took, and we'll go ahead and see if we can make sense of some of this. So when we look at our images in our data, first of all, I'd like to bring your attention to the quality of the images and how Greg was able to get a really nice representation of the plate, and then he cropped it to maximize the plate, right, so that we can have these as large as we can see them. Okay, so if we look at just the quality of the images, one of the problems with using the background I did is you kind of see the speckled nature. Well, on the plate, it almost looks like all these little speckles could be bacteria, right? Now we know they're not because we can see the background. So in this case, that's fine because I can still say, oh look, there's a colony, there's a colony, there's a colony, there's a colony. And when you take a picture at an angle, sometimes there's little shadows. So see how there's this darkness, darkness, darkness? All of these are like at the 1 o'clock position, and that's because that is a shadow. And if that happens, that's totally fine, but just don't start thinking that these are individual colonies. So be very cognizant of what you're looking at here. Okay? And here we see you really want to avoid this. We were just having a hard time trying to look into the camera, plus we have all these lights in the studio, but you'll have similar types of issues. So this would be a case of, that kind of sucks, that's not good. But fortunately, all of the microbes we can still see quite nicely. And even with my blue fingers back here, we can see everything. So this, if you were to submit this, I'd say, okay, well, as long as I can see everything, I'm good to go. If you were holding it like this and there was a big glare and we couldn't see hardly any microbes, then I would not be able to assess it. But as an instructor, I can look at this and I can look at your description and I can see if what you were looking at is borne out in the data table that you enter. And then if we look over here, well, obviously we just had a huge amount of growth. Okay, so that is the quality of the images. Now let's talk about what we're actually seeing here. So the ear gives us the best example of a quadrant streak. So here's quadrant 1, right? And it's nice and spread out, which means that going back and forth, back and forth, back and forth when I was doing this paid off. This is quadrant 2. So you can see I struck out this way, and went back and pulled them out. And then quadrant 3, I went like this. So this is the only one in quadrant 3. And then this is the one in quadrant 4. In other words, this bacteria was brought out to here, and then it was pulled up to there, and then it was brought out to here. So that shows the power of the quadrant streaking in that we have individual colonies that look like they're well separated and isolated. So good job, Andy. And what we also see is that everything is not exactly the same. Most of these look exactly like the same type of microorganisms. And I would guess, based on the color of this, the size of this, and knowing about the ear, that this is probably some type of Staphylococcus species. Now this over here, this yellow colony, this, you can never tell just by color, but my guess for this is that it is a microorganism called Micrococcus luteus because they are common on skin and they are yellow. And so anyway, this is a great looking plate. And so for these, I would call these circular for the majority of them. I would then look up closely and it looks like it's entire. And the elevation, when I looked at it before putting it away, I actually saw that each one of these were a little bit convex. And if you're looking at figure 16 colony morphology, you can see that what I'm talking about. So that's one quadrant streak. If we look at the soil, this is quadrant 1, quadrant 2, quadrant 3, and quadrant 4. And the reason why there's so much growth over here is because the bacteria can swim. So when a plate is completely covered like this, where it's just like big blob material, that means that the bacteria were swimming all over the place and filled up the plate. Now look at this. Why is it that there is, it's like going around this particular colony? Why did this bacteria not grow all the way up against over this? You can see that on some, here's a colony where this mass back here grew up and all around it. So this is an individual colony that got essentially got all pushed up on by this other swimming microbe. But here, look, it stopped. There's this perimeter. That tells me that this microorganism released an antibiotic which diffused into the plate. And so when this bacteria swam over here, it was killed and so could not grow. And we call that the zone of inhibition. Yeah. And you will be doing an experiment with zones of inhibition with antibiotics later in this course. But this goes also to the point that a lot of antibiotics are formed by soil bacteria. So that's another reason why I like to look at soil when we do this lab. So anyway, and this is the plate that stunk the most. This was very, very stinky. So I put that in my data table. Stunk like sewage. Okay. In terms of the phone over here, this is quadrant 1 over here, and then we've got some out. So again, the quadrant streaking worked because we've got them all spread out. We've got various colonies. So this is a yellow colony. This is another yellow colony. If we look at this, this would be more like irregular or spindle for the shape, right? The margin is pretty flat, so I would call that entire. Whereas over here, if we look at this, I would call this irregular shape right here, this particular colony. And if I look at it up close, and I can, you know, in PowerPoint even, I can go ahead and say, well, let's go ahead and make this even bigger, right? So that I can see exactly what we're looking at here. And so we can look at it even closer up. And so for the purpose of this, we don't need to do that. But when you're looking at these really, really close, what we see is that this is another one. When I saw this with my eye, see that little dimple right there? That actually was sticking up in the middle. And this is where a picture really can't tell you. I noted this when I was looking at it. So if you've got this little like nipple-like thing in this, and you look at figure 16, what is the elevation for that? Right, so look at figure 16. The elevation for something that looks like it's got a little like nipple type thing is umbonate. So those are the types of things that I want you to put on this, in your data table. And also how much. When you've got a plate that's covered like this, you can just get away with saying something like 85% coverage. Okay? Over here you can put something like 25% coverage plus 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and count up all these colonies. So you could say 25% coverage plus 23 individual colonies. I just made up that number. Over here, you would put something like 95% coverage. And over here, since you can count all the colonies, you should count all the colonies. 1, 2, see how these are small? So this right here would be size, pinpoint, and then this one would be large, which means that then you would want to say, well, this is probably moderate, this is small. And so count them up and say, well, I've got 2 large colonies, I've got 2 large colonies, I've got 1, 2, 3, 4, 5, 6, 7, 8 moderate, and then start counting the small, etc. So that's how you fill in the data table for this particular lab. And remember, color, shape, margin, elevation, size, and smelly factor. That's what you need to do for the lab. Okay, so now that you have analyzed your 4 plates, what we're going to do is we're going to prepare for the next lab. Okay, so what we don't need are these are 3 of the plates. So what I need to do is I need to find a plate to find a colony that I want to investigate. And what I'm going to do is I'm going to investigate a colony on my phone. So if you look closely at this plate now, the colony that I am going to choose is essentially, well, anyone that I want to, but I kind of like, I'm just going to choose this big colony right there. Okay, so what I'm going to do is I'm going to go ahead and take my marker and I'm going to circle that. And that is my colony X. And actually, if you want to go ahead and do this, I should have mentioned that, do this before you take the photos. That way when you submit the photo and we look at something like this, then it's very clear what colony X was. But if you don't do it till after you take your photo, no worries. But what I'm doing is I'm signalizing to myself that this is colony X. And what we're going to do is we're going to use this in multiple labs. Okay, so what I'm going to do is I'm going to take this and I'm going to put this in broth to start out the aseptic techniques laboratory. And so if we go and we look at the Aseptic Techniques Laboratory on the computer screen, what I want to show you right now is the flow chart that I made for this course. So right now, we've just finished this section, right? Ubiquity of microorganisms, part 1 and part 2. We have our 4 plates. We took one of our plates and we circled it right here as colony X. So now I'm going to use that plate to start out this lab right here, aseptic technique and culturing microbes, where I'm going to take that colony, put it into a nutrient broth. I'm going to go ahead and put my yeast into nutrient broth, E. coli into nutrient broth, and Staphylococcus epidermidis into nutrient broth, and then incubate that 48 hours. So this is what we are about to start right now. So when we go back to the lab, the question is, okay, what do we need for this particular lab, the aseptic technique lab, right? And so for the aseptic technique lab, we're going to need our flame in a can, a lighter, our flame in our rack. We're also going to use 3 sterile pipettes, okay? And so these 3 sterile pipettes are the ones that I have mailed to you because the kit comes with pipettes that in their description, you're supposed to put it in alcohol, and they say that that's sterile, and then you reuse it for the different microorganisms. I'm here to tell you that that is a very poor way to do microbiology, and what we're not going to do together is do any kind of poor microbiology. Okay, so like when the lab manual says that the pipette is now sterile after putting it in ethanol, that doesn't make it sterile. If anything, it would disinfect it. Okay, so what we're going to do is we're going to bypass that. Each one of those are individually sterilized, and you only use them once for each type of microorganism. So in addition to these items right here, we need these 3 sterile pipettes, and we need 4 nutrient broths. Okay, so the lab comes by saying that you only need 3, but that is for the yeast, the Escherichia coli, and the Staphylococcus epidermidis, and we also need one for our colony X. Okay, so let me take a break for a quick moment, and I will go get the cultures of Escherichia coli and Staphylococcus epidermidis, as well as the Saccharomyces cerevisiae.